So today we're going to look at Chapter 25, the history of life on Earth in evolution. So the past organisms were very different from what we see that is alive now. The fossil record will show our macroevolutionary changes over large time scales. So that's what macroevolution is change over a large time scale. What we've seen is the emergence of the terrestrial vertebrates, the impact of mass extinction, and the origins of flight in birds. So the conditions on early Earth made this origin of life possible. When we look at the chemical and physical processes on the early Earth, there may have been conditions that have produced very simple cells through a sequence of stages. So first was abiotic synthesis of small organic molecules. Second, these molecules joined together into the macromolecules. Third, these molecules were packaged into protocells. And then the origin of the self-replicating molecules. So when we look at the synthesis of the organic compounds on the early Earth, the Earth was formed about 4.6 billion years ago, along with the rest of the solar system. So the early Earth was bombarded by rocks and ice, and it likely vaporized the water. And this prevented the seas from forming before about 4 billion years ago. Earth's early atmosphere likely contained water vapor and chemicals released by volcanic eruptions, such as nitrogen, nitrogen oxides, carbon dioxide, methane, ammonia, and hydrogen. In the 1920s, A.I. Oparin and J.B.S. Haldane hypothesized that the early atmosphere was a reducing environment. And then in 1953, Stanley Miller and Harold Urey conducted lab experiments that showed that the abiotic synthesis of organic molecules in a reducing atmosphere is possible. So despite the fact that showing that it was possible in a reducing atmosphere, the evidence is not yet convincing that the early atmosphere was in fact reducing. So instead of forming in the atmosphere, the first organic compounds may have been synthesized near volcanoes or in deep sea vents. So the Miller-Urey type experiments demonstrate that the organic molecules could have formed with various possible atmospheres. Amino acids have also been found in meteorites, which are an indication of early life. So when we look at the abiotic synthesis of the macromolecules, RNA monomers have been produced spontaneously from simple molecules. The small organic molecules polymerize when they are concentrated on hot sand, clay, or rock. So, looking at protocells, the replication and metabolism are key properties of life, and may have appeared together in the protocells. Protocells may have formed from a fluid-like vesicle that had a membrane-like structure. In water, lipids, and other organic molecules, you can spontaneously form vesicles with a lipid bilayer. Adding the clay can increase the rate of vesicle formation, and these vesicles will exhibit simple reproduction and metabolism and maintain an internal chemical environment. So this chart here indicates the relative turbidity, which is an index of the vesicle numbers. So when we look at the time in minutes going across, here when you had the precursor molecules only, here you could see that you had a greater number of them when you had the precursor molecules plus the clay. Here it's showing the reproduction of these, and then the vesicle boundary and the absorption of RNA. So your first genetic material probably was RNA, not DNA. RNA molecules called ribozymes have been found to catalyze many different reactions. So for example, ribozymes can make complementary copies of short stretches of RNA. So by having it be RNA, it could act as the enzymes as well as carrying a genetic code at the same time. Natural selection has produced self-replicating RNA molecules. The RNA molecules are more stable And so the ones that were more stable would have replicated more quickly and would have left the most descendant RNA molecules. Your early genetic material might have formed from an RNA world. So these vesicles containing RNA are capable of replication, and these would have been the protocells. This RNA would have provided the template for DNA, which is a more stable genetic information. So we can use the fossil record to document the history of life on Earth. It reveals changes in the history. Sedimentary rocks are deposited into the layers called strata, and these are the richest source of fossils. Our fossil records show changes in kinds of organisms on Earth over time. So this is looking at time from the present back to 35,000 years ago, and the depth down. So we can see the time frame of when you had these different ancestors going all the way back. When we look at this, we have to keep in mind that few individuals have fossilized and even fewer have been discovered. So our fossil record is going to be biased in favor of species that existed for a long enough period of time to actually become fossils, were abundant and widespread, making them more likely to be fossils, and had hard parts that were capable of becoming fossils. So the sedimentary strata reveal the relative ages. The deeper it is, the older it is. The absolute ages of the fossils can be determined using radiometric dating. A radioactive parent isotope decays into a daughter isotope at a constant rate. So each isotope has a known half-life, the time it is required for half of the parent isotope to decay. So here you can look and see the fraction of the parent isotope remaining and the time in half-lives. So as you progress down here, you have half of the parent remaining, another, that half of it remaining, that half remaining, and half of that remaining. And each one accumulates more of the daughter isotope. So when you look at the relative amounts of daughter isotope to parent isotope, it can give you relative time frames and ages. So radiocarbon dating can be used to date fossils up to about 75,000 years old. For older fossils, some isotopes can be used to date the volcanic rock layers above and below the fossil, and then extrapolate the age from there. So when we look at some of our newer groups of organisms, the mammals belong to a group of animals called the tetrapods. The evolution of the unique mammalian features can be traced through gradual changes over time. So here, if we look where you had the tetrapods coming off, we can look early at this hinge-type jaw, a new hinge, and then a very late one with this type of hinge-type jaw. So this just allows us to do the comparisons and look at how things changed as we've moved into the very late modern mammals. So there are some key events in life's history that would include origins of unicellular and multicellular organisms and the colonization of land over time. So our geologic record can be divided into the Hadean, Archean, Proterozoic, and Phanerozoic eons. The Phanerozoic eon includes the last half-billion years, and it's divided into three areas: the Paleozoic, Mesozoic, and Cenozoic. So here you can look at your Hadean, Archean, Proterozoic, and Phanerozoic, and how we subdivide this one into these three here. This is actually making it a huge chunk of advancements that would come on through evolution. So this diagram puts things in the perspective of being like a clock and seeing the relative time frames of how long ago these different things occurred. So you can look and go backwards this way and see how long these things have been on Earth. We can see here humans are just a small little hash mark, the colonization of land, development of animals, multicellular eukaryotes, single cellular eukaryotes, atmospheric oxygen, and the prokaryotes. So the first single-celled organisms, when we look at the oldest known fossils, are the Stromatolites. These are rocks formed by the accumulation of sedimentary layers on bacterial mats. These date back to 3.5 billion years ago. Prokaryotes were the Earth's sole inhabitants for more than 1.5 billion years. So when we look, here is where we are today. This is how long back the prokaryotes go. Most of the atmospheric oxygen is of biological origin, meaning that something else that was alive had to make it. Oxygen is produced by oxygenic photosynthesis, reacted with dissolved iron, and then precipitates out to form banded iron formations. So here we can see this is how long we've had oxygen present in the atmosphere. By about 2.7 billion years ago, oxygen began to accumulate in the atmosphere and rusting iron-rich terrestrial rocks. So that was other evidence of seeing the iron-rich terrestrial rocks with the rusting. This oxygen revolution from 2.7 to 2.3 billion years ago caused the extinction of a lot of prokaryotic groups. Some of the groups survived and adapted using cellular respiration to harvest energy. So here's our time in billions of years ago. Here was when we would have the oxygen revolution, when you can see the percent of present levels that we have of atmospheric oxygen, and it going up here. So the oldest fossils of eukaryotic cells date back to 1.8 billion years ago. The eukaryotic cells have a nuclear envelope, mitochondria, an endoplasmic reticulum, and a cytoskeleton. The Endosymbiont Theory proposes that the mitochondria and plastids, which are chloroplasts along with other related organelles, were formerly small prokaryotic cells that lived within a larger host cell. An endosymbiont is a cell that lives within a host cell. So this here shows the relative time frame that we've had the single-celled eukaryotes. The prokaryotic ancestors of mitochondria and plastids probably gained entry into the host as an undigested prey or internal parasite. In the process, they became more interdependent on the host, and the endosymbionts would have become single organisms. Serial endosymbiosis supposes that the mitochondria evolved before the plastids through a sequence of endosymbiotic events. So here, if we look, we've got our ancestral prokaryote here with some cytoplasmic DNA and a plasma membrane. So here we can see it started to develop a nucleus, putting a membrane around it. And then we start to see the endoplasmic reticulum forming from that nuclear envelope. Here it would have engulfed an aerobic bacterium. That aerobic bacterium would have eventually become the mitochondria. So this is an ancestral heterotrophic eukaryote. Some would have engulfed photosynthetic bacterium. And so here you could see, along with the mitochondria being engulfed, and this would be the plastids, we ended up with the ancestral photosynthetic eukaryote. So there is a lot of evidence that supports the endosymbiotic origin of the mitochondria and plastids. Their inner membranes are similar to the plasma membranes of prokaryotes. Their division in DNA structure is similar to the organelles and some prokaryotes. So they can transcribe and translate their own DNA, and their ribosomes are more similar to prokaryotic ribosomes than eukaryotic ribosomes. So this evolution of the eukaryotic cells allowed for a greater range of multicellular organisms. We had a new wave of diversification occur when multicellularity evolved, and this gave rise to the algae, plants, fungi, and animals. So our oldest known fossils of the multicellular eukaryotes that can be resolved taxonomically are the small algae that lived about 1.2 billion years ago. There are older fossils dating back to 1.8 billion years ago that may also be small multicellular eukaryotes. The Ediacaran biota were an assemblage of larger and more diverse soft-bodied organisms. They lived from 600 to 535 million years ago. So here we can see this is the origin of the multicellular eukaryotes, so how relatively recent they are on the clock. The Cambrian Explosion refers to a sudden appearance of fossils resembling modern animal phyla in the Cambrian period from 535 to 525 million years ago. Few of the animal phyla appear even earlier, such as the spongians, cnidarians, and the mollusks. The Cambrian Explosion provides the first evidence of predator-prey interactions. So here we can see the animals were getting very recent. So here we can look at the Proterozoic and the Paleozoic, at these different times when you would have organisms forking off. So here you can see some of the sponges and the cnidarians in these early branches, even some of the mollusks. And then we have the echinoderms, chordates, annelids, and arthropods coming in a little bit later. So DNA analysis suggests that the sponges and the have a common ancestor to several other animal phyla that evolved 700 to 670 million years ago. The fossil evidence of early animals dates back to 710 to 560 million years ago. Molecular and fossil data suggest that the Cambrian Explosion had a long fuse, which means it took a while to actually get started. So the colonization of land gave rise to many more organisms because they had so many more places to live. So fungi, plants, and animals began to colonize land about 500 million years ago. The vascular tissue in plants would transport the materials internally, and that appeared by 420 million years ago. That allowed the plants to get significantly larger. So here we can see colonization of land. So the plants and fungi likely colonized together. That would explain a lot of their symbiotic relationships that we have now. Fossilized plants showed evidence of mutually beneficial associations with the fungi, the mycorrhizae, that we still see today. So the arthropods and tetrapods are the most widespread and diverse land animals. Tetrapods evolved from the lobed-finned fishes around 365 million years ago. The human lineage of the tetrapods evolved around 6 to 7 million years ago. And modern humans originated only 195,000 years ago. So the history of life on Earth has seen the rise and fall of many groups of organisms. So we've seen a lot of organisms be here for relatively short times. The rise and falls of the groups depends on speciation and extinction rates within the groups. So here you can see common lineages of ancestors A and B, 4 lineages A and B. So here we ended up with where they died out and only one going on to survive. With the lineage B, we can see only one died out and most of them went on to survive. So plate tectonics has influenced this. The land masses of Earth have formed a supercontinent 3 times over the past 1.5 billion years. We saw one 1.1 billion years ago, 600 million years ago, and 250 million years ago. According to the theory of plate tectonics, the Earth's crust is composed of floating plates on the Earth's mantle. So here you would have the crust, the mantle, the outer core, and then the inner core of the Earth. So these tectonic plates move slowly through the process of continental drift. Your oceanic and continental plates can collide, separate, or slide past each other. These interactions between the plates are going to be the cause of the formation of mountains, islands, and earthquakes. Even looking at the Earth today, we have new islands form and disappear. We have earthquakes, and we even have mountains changing their elevation as they grow and shrink down. So this is showing here where our different plates are. So here we have the Juan de Fuca plate, Cocos plate, the Pacific plate, and you can see them in various other parts of the world. So consequences of this continental drift, we had the formation of the supercontinent Pangaea about 250 million years ago, and this had many effects. This deepened the ocean basins, you had a reduction in shallow water habitat, and we ended up with a colder and drier climate inland. So here, this is showing this supercontinent Pangaea here. 135 million years ago, you can see where we had the Laurasia and Gondwana land masses that would separate. Here are the present-day continents, and here is where you would have the collision with India with Eurasia. So this is starting to look a lot more like what we have today. So continental drift has many effects on living organisms. The continent's climate can change as it moves north or south, and the separation of land masses can lead to allopatric speciation. Even if these things are slow, they do have an effect. The distribution of the fossils in living groups reflects the historic movement of the continents. So when we look, for example, the similarity of fossils found in parts of South America and Africa is consistent with the idea that these continents used to be attached.
And you have to have a certain...
So mass extinctions have occurred numerous times. The fossil record shows that most species that have ever lived are now extinct. And the extinction can be caused by changes to a species' environment. At times, the rate of extinction is increased dramatically, and when this happens, we call it a mass extinction. It's the result of disruptive global environmental changes. So we have 5 big mass extinction events where 50% or more of the marine species became extinct. So we can look at the time in millions of years ago, and you can see where there are these points in here when we look at the number of total extinction rates over the years. So the Permian Extinction defines the boundary between the Paleozoic and Mesozoic eras of 251 million years ago. That mass extinction occurred in less than 500,000 years, and it caused the extinction of about 96% of the marine animal species. A number of factors may have contributed to these extinctions. One would be the intense volcanism in what is now Siberia. We've seen global warming and ocean acidification that results from the emissions of large amounts of carbon dioxide from the volcanoes. So volcanoes have been influencing the environment for years, well before we were here. And then the anoxic conditions resulting from the nutrient enrichment of ecosystems. The Cretaceous mass extinction occurred 65.5 million years ago. Organisms that went extinct include about half of all the marine species and many terrestrial plants and animals. This included most of the dinosaurs. The presence of iridium in sedimentary rocks suggests that a meteorite impacted about 65 million years ago occurred. The dust clouds caused by that impact would have blocked out sunlight and altered the global climate.
Lab days are Tuesdays and Thursdays.
The Chicxulub Crater off the coast of Mexico is evidence of a meteorite that dates back to the same date and time. So here you can see where this crater would be, right off of Mexico.
Getting all their labs done, then we can get out here early.
So it's estimated that we may have a 6th mass extinction underway. It's estimated that the current rate of extinction is 100 to 1,000 times the typical background rate. We see extinction rates tend to increase when the global temperatures increase, so data suggests that a 6th human-caused mass extinction is likely to occur unless dramatic action is taken. So here we can look at the relative extinction rates of the marine animals, and we can see here where the mass extinctions occurred. And when we look at the cooler relative temperatures versus warmer relative temperatures, 4 of them are on the side of warmer relative temperatures. Consequences of mass extinctions, they can alter the ecological communities and the niches available to the organisms. It can take from 5 to 100 million years for the diversity to recover after a mass extinction. So a mass extinction can change the types of organisms found in ecological communities. For example, the percentage of marine organisms that were predators increased after the Permian and Cretaceous mass extinctions.
The bookstore sells it, so does Amazon, so does Walmart.
So here we can see the percentage of predators looking back in the time millions of years ago. So here, after these mass extinctions, Permian mass extinction, we see a peak and an increase in the predators.
Anywhere from $74 to $12.
So lineages with novel and advantageous features can be lost during mass extinctions. By eliminating so many species, the mass extinctions can pave the way for adaptive radiations to occur. Adaptive radiation is the rapid evolution of diversely adapted species from a common ancestor. So these will tend to follow mass extinctions, the evolution of novel characteristics, or colonization of new regions. So mammals underwent adaptive radiation after the extinction of the terrestrial dinosaurs. So a big predator was out of the way and allowed them to flourish. The disappearance of the dinosaurs, except the birds, allowed for the expansion of mammals in diversity and size. So other notable radiations would include photosynthetic prokaryotes, large predators in the Cambrian time, and land plants, insects, and tetrapods.
$3 mask. It's about $20 minus the book, about $20 worth of little stuff right there.
So here when we look, we can see some of the adaptations here on land with the monotremes, marsupials, and eutherians. So
in the summer, and if they change the process, you won't have to worry about
much greater numbers of the placental mammals.
Like what I have here up on my desk.
So adaptive radiations can occur when organisms colonize new environments with little competition. An example of this are the Hawaiian Islands. They're one of the world's greatest showcases of adaptive radiation. So here we can look at the different islands and the years over how they have developed and adaptive radiation that we've seen in these different plants. So here from Kauai, from Oahu, Molokai, Lanai, down here, Maui, up here, and Hawaii.
Again, in the summer, they may have changed the amount and actually charge me more.
So studying genetic mechanisms of change can give us some insight into large-scale evolutionary change. Genes that program development control the rate, timing, and spatial pattern of changes in an organism as it develops into an adult. So heterochrony is an evolutionary change in the rate or the timing of developmental events. It can have a significant impact on body shape. If we look at the contrasting shapes of the human and the chimpanzee skulls, these are the results of small changes in relative growth rates. So here, when you look at a human fetus and a chimpanzee fetus looking much similar, but just change in growth rate leads to quite a bit of a difference between a chimpanzee and a human adult skull.
They're all open book.
Heterochrony can alter the timing of reproductive development relative to the development of non-reproductive organs. In paedomorphosis, the rate of reproductive development accelerates compared with somatic development. The sexually mature species may retain body features that were juvenile structures in the ancestral species. So here we can see these gills. When we have the changes in spatial patterns, you can have substantial evolutionary change that can also result from altering the genes that control the placement and organization of the body parts. Homeotic genes are going to be ones that determine the basic features, such as where the wings and legs will develop on a bird, or how a flower's parts are rearranged. So Hox genes are a class of homeotic genes. These will provide positional information during animal embryonic development. If the Hox genes are expressed in the wrong location, then the body parts can be produced in the wrong location. An example, in the crustaceans, the swimming appendage could be produced instead of a feeding appendage. And that is the end of this section of notes.
I will probably have that class